I was growing bacteria that function in breakdown of urea. So then I continued to subculture and break down until I had pure cultures right here, and proceeded to study the strain both phenotypically and then eventually genotypically. So first of all, I found that I documented all of the physical traits of my bacterium, pretty basic colonies, small, margins, slightly raised, grew very fast, very easy to work with, right? And grew in the 32 degrees Celsius, and did various stains. I did a purple-violet stain, which you can see right here at 100 times. You can see my bacteria are very tiny, even as bacteria go. I also found that it was gram-negative, and that they're rod-shaped. I also found that they're highly modal. As you can see here, we have a swarming region. This is where my initial bacterium was, and as I came to find out, genotypically they use flagella. I also studied antibiotic resistance, and I found that most antibiotics that we studied showed that the bacterium was clearly, by this ring right here, was clearly susceptible to it, except for penicillin, which is interesting, and I looked further into that also in the genotypic analysis. So we extracted DNA and sent it off for the genome, studying the genes. We sent it off to NHTI, and they sequenced it for us. And we trimmed it down, used spades to assemble it, Proca to annotate it, and then looked into specific genes of interest, starting with the 16S rRNA to figure out exactly what kind of bacterium it is, and found that it was, in fact, the Shewanella bicestri, which is close to other Shewanella bacteria like Shewanella baltica and putrefaciens, which both are known to function in the breakdown of waste in the Baltic Sea and other places. So that was awesome. I knew what I had, and I wanted to study specific genes of interest, specifically genes that function in breakdown of urea, ammonia, nitrate, and nitrite. So I grepped a lot of those, looked them up, and found that there are certainly many that are present. Hydrogen urease, histamine ammonulase, things that break down, which is good. That's exciting because it supports the goal, the point. I also wanted to look into this resistance to penicillin, which is really interesting. And one major pathway for a bacteria to be resistant to penicillin is to have a beta-lactamase, which breaks down the beta-lactam ring in penicillin and other antibiotics like cephalosporin. So I found 4 genes that code for beta-lactamase. 2 are just general ones that certainly function in it but don't have a specific name. And then 2 were of particular interest because they are on the brink of, or actually known as, extended spectrum beta-lactamases, which really do play a major part in, there's a lot of interest in that because they're a big deal in human pathogenicity and treating diseases that have this. So Shewanella can be pathogenic, and if you were to contract the disease from Shewanella bicestri, that would be something to consider. So I found that, there were those genes. Beta-lactamase oxidase-10 is not ESBL, but is a precursor to it, so it could easily become that. So those are just a few of the little bit of things that I've studied genotypically. There's a lot more that I could do to specifically study and analyze. In fact, I'm in the process right now using a program called K to study different orthologs that this bacteria might match that are known to function in breakdown. Not just the presence, but actually functionality in the breakdown of urea and other nitrogen-based molecules. So for my conclusions, I definitely would have a lot more to do. In fact, there was a little bit of dilemma with the amount of genetic material. There's a little bit too much, so I would want to look into that a little further, potentially some contamination, and it could be plasmid DNA, but I just have to study that further. Some other applications, I would actually like to, if my fish ever got a disease, try to treat it with a penicillin-like antibiotic, so that I don't hurt my bacterial culture in my fish tank. So that's a unique, specific application that I can use right away. All in all, I definitely think I've made a case that Shigella bicestri does function in the breakdown of waste in my fish tank, and could use further study in order to make that point even further.